Luke, Jamp, my friends, can you guess what we are starting this week with?
Is it a nice wee review?
Is it a nice wee review?
Is there an echo in here?
Yes.
Is there an echo in here?
Is there an echo in here?
Yes.
Yes.
Yes, we are starting this week
Okay,
with
good, it is.
a
Okay.
review. So this review is from Alice.Blondie.
Ooh.
So this review says, this podcast is amazing. The perfect balance of science, nerdiness, and banter makes me realize that my A-level sciences were not a complete waste of time and that I do remember some stuff. Really recommend the Chernobyl episode.
Ooh. Oh, that was a good one.
That was a fun one.
It's an oldie.
It was an oldie, but a goodie. Exactly, bang on. If you would like your review read out at the start of this podcast, leave us a 5-star review on Apple Podcasts and we'll read it out for you. I said those words a lot of times. Let's start the show. Hello and welcome to the SCI Guys, the show where we talk about the crazy, weird, and wonderful stories from the science world. I'm Corey, and as always, I'm joined by my co-hosts, Jamp and Luke Cupford.
Hi there.
Hello.
This week, we're talking about miraculous molds and accidental accomplishments.
Ah.
Oh. You guys have an of understanding.
Guys, are any of you an accidental accomplishment?
Couldn't quite call myself an accomplishment.
I don't know. An accidental accident.
An accidental nuisance, perhaps. But I'm sure we've all heard of antibiotics, but have you heard of penicillin? I'm sure you have.
Yes.
Do you guys know the story behind penicillin and its discovery?
It was an accident.
Yes, it was.
It was an accident.
That's right. I wonder where you got that from.
That's all I know about
So
it.
you know. Fair enough. Well, that's what we're talking about today. Penicillin, its discovery and its production and how it changed the world. Shall we dive right in?
Immediately.
What is penicillin?
It's an
medicine.
antibiotic.
That's the exact first line of my notes. That
was
is
gonna
right. Penicillin is an antibiotic.
I was going to say that, but I double guessed myself. So I was like, medicine.
It
Both
comes
are right.
from a fungus, does it not?
It does, yeah. We're going to get right into that. The
Ah.
fungus that it comes from, one of them is Penicillium notatum.
Where did they get the name from then? I wonder.
Shut up, Vince.
Is it someone's name?
So it was discovered in 1928 by, do you guys know who it was discovered by?
See, I was going to say Ian
Mr.
Fleming, but then I
Penicillin.
realized he wrote the James Bond books.
He did write the James Bond books. You're close. You've got
Yeah.
one of the names, right?
Fleming.
Yep. So it was Alexander Fleming.
Ah, yeah.
Who is a Scottish, I say is, he was a Scottish scientist. Yeah, and so in 1928,
Take it, he's dead.
yeah,
Okay.
yeah, well, he
No,
discovered,
actually, he dropped his Scottish passport, James.
He got married. Changed his last name.
yeah, he discovered penicillin in 1928. I
Yeah.
feel like he's almost certainly dead by
And
now.
he just recently got married. Oh.
Yeah, congratulations, Alexander, way to go.
And
Mr.
reneged on
Penicillin.
his Scottish citizenship at the same time.
So yeah, in 1928, he discovered penicillin. But we'll get into this later, but it was actually two other scientists, Howard Florey and Ernst Chain, who developed it further so that it could be used as a drug. So what is penicillin? Do you guys know? We've said it's an antibiotic, but what are antibiotics?
Thing that kills
Biotics.
biotics, yeah. Thing that kills
So
infections.
Well, if you say it kills infections, that's not technically true because you can be infected by you can be infected by a lot of things. You can
Kills
be-
By a
bacteria.
virus.
Yeah, so you can have a viral infection, you can
Yeah.
have a fungal infection, or you can have a bacterial infection. Now, antibiotics kill bacteria. Or not necessarily kill bacteria, but they're chemicals, specifically small molecules, that enter and basically get involved with important parts of a bacterium, and basically interrupt its ability to survive or reproduce.
Mm.
Ah.
So, in short,
So just a big cock block.
if bacteria had those, then yeah, sure.
Antibiotics are the cock block.
Yeah, so antibiotics are basically just drugs that we use to kill or stop the growth of bacteria. So obviously not all bacteria are susceptible to all antibiotics, but if they are susceptible to an antibiotic, they will either stop growing or die when subjected to it, which is pretty cool.
Good.
And the important part is they only affect bacteria. So they don't affect fungi. They don't affect fungi or viruses, and they don't affect human cells or mammalian cells. They don't affect anything that isn't a bacterium.
That's good for us because we are mammals.
Yes.
But do they affect the
knowledge.
bacteria in us, though, don't
Yes,
they?
they'll affect the bacteria in the gut, which is why actually when you take antibiotics, you could have stomach issues, you could have a lot of problems because as we've covered in our microbiome episode,
There's good bacteria and bad bacteria.
there's bacteria.
Essentially.
Yeah, there's bacteria in you that you need to survive. So when you take antibiotics and they affect those bacteria, you're killing the good guys while getting rid of the bad guys, you know?
Yeah.
But yeah, they can also interact with other things like alcohol. There's actually only a couple of don't take my medical advice, but there's actually only a couple of antibiotics that interact really badly with alcohol. So even when on the packet it says don't mix with alcohol, if you Google it, it will tell you the list of like basically 2 antibiotics that actually interact with alcohol quite badly. Yeah. Anyway.
Will it affect how well the antibiotic works in any way? Like slow it down?
Generally, no.
Wow.
Weird.
No, I mean, look,
Strange advice then.
I wouldn't. I think it's more of a precaution. I wouldn't say if your doctor advises you not to drink on antibiotics, then don't drink on antibiotics. Obviously, listen to your doctor. But most people have the kind of misconception that you can't have alcohol on any antibiotics. When realistically, you technically can.
Yeah.
It won't necessarily hugely affect the antibiotic, but obviously, drinking alcohol affects your immune system. So if you're not well, you probably shouldn't be drinking anyway.
True.
I see.
Yeah. So
Who gets drunk when they're ill?
I had
Quite a lot
You
of
know what
people,
this calls
James.
for?
my
Well, he does.
My 19th birthday, I had what's the tonsillitis that just would not clear up. So I had to go to the doctor to get antibiotics. And yes, I got very drunk on my birthday while on antibiotics.
Oh, but it's your birthday. That's an exception,
Yeah,
right?
yeah, it's allowed.
Yeah.
It's allowed. You can do whatever you want
You're
on your
allowed.
birthday. So the important parts of the bacteria that the antibiotics target are usually the proteins or the sugars in the bacterial wall, or important enzymes that the bacteria use to kind of replicate.
Mhm.
So when an antibiotic sticks to those important parts of the bacteria, they'll either like sort of stop them from working, sort of disable them, or just completely destroy them. So when that happens, as I've said, the bacteria either stops growing or just dies. So when it stops growing, that's a bacteriostatic effect, and when it dies, that's a bactericidal effect. Bactericidal. Yeah. So how does penicillin work specifically? Do you guys know? I don't. I wouldn't expect you to know.
It's a 50-50 chance, isn't it?
Oh well. Okay. So generally, this is something I should mention. The bacteriostatic effect and the bactericidal effect. That doesn't mean that some antibiotics have each effect. What it means is if there's, say, less of the antibiotic, it will have a bacteriostatic effect. And if there's more, it'll have a bactericidal effect.
Okay.
Yeah.
Right.
So if you increase the concentration, you can kill more bacteria effectively. If you think about it, if you're only taking out, say it's a
Makes sense.
yeah, say it's an antibiotic that takes out part of the cell wall. If you take out a few bricks from the wall, it's not necessarily going to crumble. But if you get 10 more guys that start taking out all the bricks, that wall's falling down.
So it's like if you drink a lot of alcohol, you become impotent. But if you drink even more alcohol, you die.
You do. Yeah, alcohol
There
poisoning.
you go. That's how we remember it.
So bacteria gets alcohol poisoning, essentially.
Yeah.
Look, that
And
is
dies.
that's absolutely spot on.
Thank you.
A little bit makes them drunk. Makes the bacteria drunk. A
Make
lot.
them a bit floppy.
Alcohol poisoning have to get the
Doesn't
stomach pumped
work.
or they die.
Can't get it up. And
Yeah.
then that's the bacterial static effect. And then the bactericidal effect is when you just sadly pass away.
You get taken to bacteria hospital, and they have
Yeah,
to
I did
pump
that.
your
I did
bacteria
that
stomach.
at my 15th birthday party.
Oh.
I went to hospital drinking too much alcohol.
Oh, lovely.
Bacterial static effect.
Okay,
What
moving
a
on.
happy anecdote.
Moving on. Penicillin works by weakening the bacterial cell wall. So what it does is it stops an enzyme called transpeptidase from working. Now that enzyme builds and repairs. That enzyme builds and repairs the bacterial cell wall. Now the bacterial cell wall is made up of this thing called peptidoglycan. Yeah, have you heard of this before?
No.
Yes.
You have?
No.
Okay.
You lie.
So what peptidoglycans do is they create a mesh sort of structure around the membrane of the bacteria.
Yeah, Corey, I know.
So membrane is kind of like just the normal barrier between the inside of the cell and the outside of the cell.
Mhm.
And then there's a cell wall on the outside of it, which is like a big tough barrier. It's the difference between having, say, kind of just normal skin and then
Right.
having, say, calluses or a suit of armor.
Ooh.
Yeah?
Tough.
So our cells don't actually have a cell wall. We've only got a cell membrane. And other cells, so like plant cells have got a cell wall. These bacterial cells have got a cell wall as well. So again, made up by peptidoglycans. What penicillin also does, it activates an enzyme in the bacteria called autolysin. And what that does is that cuts open parts of the cell wall. The enzyme's normally only activated when the bacteria multiplies. So when the bacteria multiplies, that enzyme will cut open the cell wall so that the bacteria can split off into a new bacterium.
Right.
But what this antibiotic, what penicillin does, is effectively it stops the enzyme that makes new cell wall, and it kickstarts the enzyme that breaks up the cell wall. So it basically uses the bacteria's own enzymes against it to make it just kind of shred itself apart.
Oh, okay. That kind of makes sense.
Yeah, I can go over it again.
No. This biology stuff is just my worst area.
It's big words for a really simple concept.
Yeah.
Basically,
Yeah.
I mean, penicillin goes in and it makes the bacteria start breaking down its cell wall
Yeah.
faster than it's building it back up again.
Yeah.
And it's
Yes,
that
okay,
simple.
Cool.
Like when Professor Quirrell crumbles away at the end of Harry Potter 1.
Not unlike that.
Yes.
The bacteria are like.
Yeah,
yes.
crumbling away.
Harry Potter is
What is
penicillin.
this magic?
Look, Harry Potter is penicillin and Quirrell is a bacterial cell wall.
And Lily Potter is Fleming, who made penicillin.
Sure.
Yes,
Okay.
sure.
We're getting ahead of ourselves.
Hell yeah. That's how
we
we're
can
going to
keep this
remember it for the
going.
quickfire round, James.
So here's the important part about cell walls in bacteria. What they do is they make it stronger. Like I said, it's kind of like callous skin or a suit of armor
Yeah.
around the membrane. So it makes the bacterial cell stronger.
Yeah.
But when you get rid of that, it still has its membrane, so it doesn't just split apart. But what happens is water can get in. So a bunch of water starts rushing into the cell, and the cell just pops.
Like a bubble.
Osmosis.
Yes, exactly. Osmosis. And
Like a little bubble.
yeah, it pops. It's like overfilling a water balloon, and it just
Wow.
goes boom.
Ah.
That's really
Out of
horrible for the
existence.
cells, isn't it?
Yeah, it is. It is quite horrible. Thankfully, we can't hear bacteria scream.
That's horrible. There was a guy once who was asked, I can't remember who it was, and he was asked, he was like vegan, and he goes, someone asked him, like, you know, when you eat plants, you know that they feel pain too. And he replied, yes, I know, but I can't hear them scream.
Amazing.
This is true.
Yeah, so I've kind of explained that. You guys got your heads wrapped around how penicillin works?
Yeah.
Yeah.
Yeah?
Surprisingly.
Yeah, I mean,
Biology's not my strong suit, but
I think the biggest
it seems fairly simple.
Yeah, I mean, I think the biggest thing with a lot of things in biology is they've got super long names, which makes it
Yes.
hard to wrap your head around.
That's what throws me off.
But a lot of these things,
But
once
it's
you
really easy.
once you understand what things do,
Mm-hmm.
proteins usually have like one function.
Mm-hmm.
So once you understand that, it's really straightforward to know what's going on.
Yeah.
If it turns on the thing that builds it,
Yeah.
if it turns on the things that that destroys it and turns off the thing that builds it, you can figure out what's going to happen.
Yeah.
You know, it's gonna
A
break down
little
the cell.
switch.
So that's how penicillin works. Now, on to Alexander Fleming, our lovely friend, Mr. Fleming. Now, he was
Yes.
born in Ayrshire, in Scotland, on August 6th, 1881, which
is a palindrome.
before Luke is what?
113 BL.
That is
Damn.
correct. So he was a bacteriologist and he's also a doctor. So kind of early on in his medical career, he became interested in the sort of natural bacterial action of blood and in antiseptics.
Ooh.
So he lived through World War One, and he was a doctor in World War One.
Mhm.
So he kind of throughout his military career, he continued studying so sort of bacteria and that whole kind of area. And then at the end of that, he kind of decided to work on antibacterial substances, specifically ones that wouldn't kill animal cells.
Mhm.
So obviously the difficulty is if you're trying to kill a bacterium, how do you kill it without killing your own cells, because you're made of cells just as much as the bacterium is. Yeah, there's a different structure and there's a lot of different things going on, but there are things that will basically kill cells in general. And it's finding something that kills specific cells rather than all cells. So that's what he set to work.
Injecting bleach into your lungs.
Don't do that.
Exactly.
Where did that come from? Oh, did it come from Trump?
Yeah, no,
Yes.
I don't even want to talk about that. I don't want to talk about it. That's awful.
Would kill the bacteria.
It would kill you.
The virus.
It would kill your lungs.
You too.
It would kill the whole entire person.
I mean, if you're
Not
gonna
totally wrong.
if you're gonna
Just
do
the
that,
person wouldn't survive as well.
if you're gonna do that, you may as well just jump into a fire.
I was going to say, do you know throwing yourself off a cliff also kills the virus?
It's like, oh no, my hands are dirty. They're covered in bacteria. I know what I'll do. I'll stick them on the hob. Sure. Okay. Your skin can't be dirty if it's burned off, but then you don't have skin.
See, Corey, we are too quick to criticize this stable genius.
It does check out.
That's the equivalent of someone going for chemo, right? And instead of just trying to target the cancer cells, just saying, why don't we just cut off everything? Just get rid of all the cells.
Just amputate everything. We'll make
However,
7 individual people.
Okay, here's my one thing where I'm like, I wonder what you think of this, Corey. I reckon. Okay, before I say this,
I am so worried to hear what you're about to say.
Before I say this, I do not think this is what Donald Trump meant.
It needs a disclaimer.
Right? This is not what I think Donald Trump meant. However, I do wonder whether every any molecule, any kind of structure at all, surely has some kind of natural frequency, right? And if you could create a specific enough frequency of radiation, could you not cause an individual virus particle to resonate in such a way that it broke apart? Would that not work?
I would hazard a guess at no. Generally, what radiation does is it tears apart DNA and it tears apart cells and it stops them from being able to replicate. So in the case of targeting radiation specifically to hit a virus, that wouldn't quite work. Otherwise, we'd be doing that with cancer. So what we do with cancer right now, chemotherapy is and radiation treatment. So what radiation treatment is effectively firing radiation at one specific part of your body that's got cancer in it and trying to limit the damage to the cells around it.
Yeah.
If we find a way to target specific kinds of cells, then cancer's not a problem anymore. Because the issue with cancer is trying to kill specifically cancer cells without killing the cells around them. So if we were able to do that, then we've cured all infections and all diseases.
I just was trying to watch Trump, and I was like, I'm always trying so hard to find somebody who I think is like so stupid, and be like, okay, well, might they have a point? Like, are we so arrogant that we're just like rule out everything that anyone ever says? And I feel like that is mostly the case with Donald Trump. Is most of the stuff is just ruled out immediately with him because he's a bloody idiot a lot of the time.
Well,
I was basically looking at it going, well, might there be some accidental point that he's made here? But this is the same thing as what I said to Josh once on Educating Josh, where I said to him, interestingly, the Earth could be round in 4 dimensions and round in 3 dimensions and flat in 4 dimensions. And he was like, yes, Luke, but I don't think that's why they think it's flat.
Well, you've actually brought an interesting point up there. You're saying you try and give people the benefit of the doubt. If they say something that may seem idiotic, you try and look for the truth in it, because there might be some. And actually, that's quite relevant. What you're kind of getting to there is accidentally being right about something, which brings us back to our episode, because Alexander Fleming accidentally discovered penicillin. So Alexander Fleming, as we were talking about, he discovered penicillin in 1928. So I was telling you about how he studied sort of bacteria for a while. He was in the army. Now, this is something that I think is quite interesting. Whenever someone discovers something, we often think, yes, that's the one thing they've done, and they've done nothing else in their career. Their entire
rest
career
of time, that's what they're remembered for.
Exactly. Their entire career led up to this point, and that was it. And you forget that just because Alexander Fleming discovered penicillin, it doesn't mean that he didn't make any other groundbreaking discoveries in his life. So in 1921, he discovered basically this important sort of antibacterial substance called lysozyme. Now, he discovered that in I think he discovered it in snot. It's present in tears. It's present in your body. You produce lysozyme. It's also present in egg albumin. So it's a natural sort of protein enzyme that a lot of different things produce to have an antibacterial effect. It breaks down the cell wall of bacteria. So yeah, I mean, this is the thing. He discovered lysozyme, which again, as I've said, is in egg whites and stuff. Which is it's incredible that he not only discovered penicillin, but he also discovered other things about sort of antibacterial. Yeah, in the antibacterial space that we don't really think about. Because I'm sure if anyone's got any kind of cursory knowledge of Alexander Fleming, they'd think, yep, this is the one thing he's done.
Penicillin.
Yeah, I just think it's interesting to point out. Scientists have long lives and do a lot of stuff.
They do.
Yeah. After he discovered lysozyme, he kept on working, and I've got a quote here from him. When I woke up just after dawn on September 28th, 1928, I certainly didn't plan to hold on, he's Scottish. When I woke up just after dawn on September 28, 1928, I certainly didn't plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer. But I guess that's exactly what I did.
Why are you putting on a Scottish accent when you are Scottish yourself?
People don't think I'm Scottish.
Do they not?
Well, it's not quite strong enough, is it? People just ignore it.
I think it's strong enough.
Thank you. Thanks, yeah. I mean, look, I'll put on a stronger accent if I have to. If I need to. If I want to get into character.
Are you putting one on now?
No, this is just mine.
Oh, okay. I can never tell.
This is the thing that's happened. Since moving down to England, my accent is softened because when I moved down, English people didn't understand me.
You have to communicate with people.
Yeah.
And they don't understand you if you're too strong.
Exactly. And it's been toned down completely subconsciously. As soon as I talk to someone Scottish, it's right back up. As soon as I go back to Scotland, it's back to normal.
I'm the same with Australia.
Yeah.
When I go back to Australia, it's more Australian. When I'm here, it's more not Australian.
I think when you talk to someone that's Australian, or in my case Scottish, that you automatically switch back to your normal accent. It's always interesting hearing someone with a regional accent on the phone to say their parents.
Yeah.
And they suddenly switch back and you're like, oh.
It's really strong.
So yeah, he revolutionized medicine with this discovery. And I'll just skip to the end of his life. He died on March 11th, 1955, and he's actually buried in St. Paul's Cathedral.
Oh.
Yeah.
That's kind of close.
So, penicillin, how was it discovered? So he was working on the influenza virus in 1928. And apparently he went away for a while and he came back. I think he went on holiday supposedly and came back. But I've not seen that in a lot of sources, just in one place. So he could have not gone on holiday. But what we do know happened is that he noticed that mold had grown in one of his petri dishes. So he had this petri dish to grow a bacterium called Staphylococcus. Now, he noticed that the mold that had grown on the plate, there was actually a ring of bacteria-less area. So around the mold, there wasn't any bacteria.
Oh.
But there was bacteria in the rest of the plate, which was interesting because you wouldn't expect there to be a very clear space between the two. So he initially called this substance that the mold was producing mold juice. Because while he was a preeminent scientist of his time, he was terrible at naming things.
I want that to be Corey's new nickname. All right, mold juice.
Mold juice. I was going to say petition to call penicillin mold juice from now on.
So he studied this a little bit more, and he found out that this mold, if you grew it, you could stop the growth of the bacterium Staphylococcus, even when you diluted it 800 times.
Whoa.
Yeah. Which is a lot. Which is insane. Yeah. So he named the active substance in this penicillin. So the active substance is just the thing that does stuff. So what happened when it was discovered and what was it used for? So I told you about Dr. Ernst Chain and Dr. Florey. These two were quite interesting. So Howard Florey was Australian and Ernst Chain was a refugee from Nazi Germany. Now, what happened was that Alexander Fleming didn't have quite the money or the funding to continue researching penicillin. These two took it up, and what they did was they made kind of a series of really crude penicillin mold extracts. So they made the mold, they grew that, and then they took the fluid from it and extracted that. But the issue was that they just couldn't get enough penicillin from it. So in the summer of 1940, in the height of World War II, they had some experiments done on 50 mice, and they infected those mice with Streptococcus, which is deadly. Now, these mice, half of them died, but the other half survived because they had received penicillin injections.
All of them that received rejections.
Yeah. So all the ones
So-
that were receiving penicillin injections survived.
Wow.
And I mean, this point in history, bearing in mind, before this, if you were, say, scratched and it got infected, you
Yeah.
could die.
Yeah.
If you got a bacterial infection that was bad enough, that was it. You sign your will because you're on your way out. And they
Wasn't a
found
joke.
effectively a magic bullet that you can fire it at any not any bacteria, but you could fire it at bacteria and it will only kill the bacteria. It won't harm your cells at all. So the issue that they then had was producing enough penicillin to treat a human person. Because I'm not sure if you've noticed, but humans are slightly bigger than mice. Now, they could produce some penicillin, but getting it in a pure enough form and a high enough quantity was presenting an issue. So in September of 1940, only a few months later, they tested it on the first human. Not the first human to exist. This was the first human. This was the first human that they tested
They
on.
tested it on Adam.
No, his name was actually
I made
Albert.
so many mistakes.
Oh, close.
Yeah, so his name was Albert Alexander. He was 48 years old, and he cut his face working in a rose garden. The scratch got infected with streptococcus and staphylococcus, and that spread to his eyes and into his scalp. So he was admitted to hospital, and he was treated with the drugs of the time, so the drugs that he would usually use to treat bacterial infections. But the infection still got worse, and then he got abscesses in his eyes, in his lungs, and in his shoulder. So they heard about this, and they asked the hospital, "Look, we've got this experimental drug that we know it works. We've used it on mice. Can we treat this man with it?" And the hospital obviously accepted. And after 5 days of injections, Alexander started recovering. And this was the first man to ever be treated with antibiotics. So the issue was they only had enough for about 5 days or so. So they got him to the point of recovering, but they didn't have enough to continue treatment. So he ended up dying anyway. Yeah.
Because he then had like the more resistant ones left. Oh, that's horrible.
Let's weed out the weak ones and leave the strong.
Oh.
Yeah. So
That's why you should always
And that came
continue
from
your
his
course of
garden.
antibiotics through to the end.
that is true. You need to continue your course of antibiotics, because what's happening is each time you take the
Yeah.
antibiotics, you're cutting down the crop effectively. And you need to get that level of bacteria down to a point where your immune system can deal with it on its own. And that won't happen if you stop halfway through, because you feel better because the bacteria started to wane. But if you stop taking your antibiotics, they can just start growing again and overwhelm your immune system.
Yeah.
So you
That's
need.
so interesting that you, because I never would have thought that you are using antibiotics, you're getting the bacteria down to a level your immune system can cope with. I would have always thought that the antibiotics kills all the bacteria, but there's little stragglers left over, by the sounds of what you're saying, that are then killed by the immune system. I never knew that.
I guess it's hard to tell when you've got all of them, when you've killed it all.
Well,
Yeah,
okay,
you can't
so
see them, can you?
So antibiotics can kill basically all of the bacteria in you, but realistically, there are going to be a few that are There can be a few that are resistant to it. So,
right, okay.
so long as your immune system is kind of able to take care
And
of
you
it,
start getting better anyway before
yeah,
you're completely rid of it.
yeah. So you start getting better after a couple of days of antibiotics because
Yeah.
the bacteria have gone down to such a low level. But that doesn't mean that they've.
The worst ones are left behind.
Well, it's not even that. It's just the fact that you've not necessarily killed all of them. They can still be replicating, and if you stop taking it before you've gotten rid of them all, or gotten them down to such a low level that they're not presenting an issue, then they'll just start growing again and overwhelm you. So there was also a biochemist called Dr. Norman Heatley who was using every single container he had to grow penicillin mold and pull off the fluid, the penicillin, and kind of purify the antibiotic. So he was growing it in bottles and bedpans, anything, literally anything he could get his hands on. But they still had an issue. They still had an issue with producing enough. I mean, nowadays we use big fermentation tanks, but back then they just didn't have that to produce the penicillin. So in the summer of 1941, just before the United States got into World War II, Florey and Heatley went to the United States and worked with some American scientists to try and figure out a way to mass produce penicillin. Now, penicillin notatum or notatum, the one that Fleming initially found, that mold couldn't produce penicillin in a high enough concentration to be worth anything. It produced it, but it just wasn't enough. So they were trying to find a species of mold that could produce enough penicillin to be worthwhile, a more sort of productive species. So apparently, on a hot day, a lab assistant named Mary Hunt came in with cantaloupe, a melon, and it had this golden mold on the outside of it, supposedly.
Ooh.
And it turned out that mold was Penicillium chrysogeum, which had 200 times the amount of penicillin that the original mold had. So, a stroke of luck, they found this mold, and they basically they enhanced it with X-rays so they mutated it using X-rays so that it would produce more penicillin.
So that was a second accident.
So yeah, the melon was the second accident. The first accident being actually discovering it in the first place.
Yeah.
So yeah, and this mold, like I said, gave 200 times the amount of penicillin as the other one, but they still needed more. So they hit it with X-rays to make it mutate and produce more. And then after that, they filtered it and produced 1,000 times as much penicillin as the first batches that they were using from the previous mold. So by 1942, so I mean, a year later, they had a fully injectable form of penicillin ready for mass production, which is just in time to help World War II soldiers. Bearing in mind, in World War I, bacterial pneumonia killed 18% of soldiers.
Yeah.
In World War II, it killed less than 1%. And that's thanks to penicillin. The fact that
Did
they
both
managed...
sides have penicillin?
Sorry?
Did both sides have penicillin?
I've actually got... That's the exact next segment that I'm getting onto. So the British government really tried to stop penicillin from getting into basically the Nazis' hands. But penicillin basically leaked out. People started talking about it, obviously, and it kind of got out. Here's what happened. A Swiss company wrote to Florey trying to get some of P. natatum, the original mold that they grew penicillin from. Now, Florey contacted the British government because he wasn't too sure about what to do on this. And then they tracked down where Fleming's original cultures had been sent to, because Fleming had been sending these out. He found this sort of wonder mold that grew this bacterial killer, and he was sending it out to different labs because that's kind of what you do. You in science, if you want to get more people working on it, you can just do that. So they tracked down everywhere that it had been sent to, and Fleming wrote, during the past 10 years, I have sent out a very large number of cultures of penicillium to all sorts of places, but as far as I can remember, none have gone to Germany. Now, Florey thought...
Germany.
Well, that's the thing. Florey thought that without the mold, no one could make penicillin. But
he made penicillin.
Well, yeah, because he had the mold
From a lemon.
Oh, right. Okay.
So
something.
yeah, so if you don't have the mold, you can't make penicillin.
Right,
even though he published basically a blueprint to manufacture on a small scale. But that's the key thing. If you don't have you can have an oven, but if you don't have any flour, you can't make any bread.
Yes.
Unless you're making gluten-free bread.
You still need flour. You need gluten-free flour.
Dun, 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 dun.
I wonder what the definition of flour is.
Oh my lord!
Because I think because like gluten-free bread is often made from like potatoes and stuff.
Yeah, but you can have gluten-free flour, like chickpea flour. Gram flour is gluten-free because it comes from
but flour means a grain. So if it's chickpea, it's technically not flour.
Yes, but milk means like from the mammary glands of a mammal.
Oh yeah, but they're not allowed to use
milk.
milk. They're often not allowed to use milk anymore. It's been banned in lots of places. Soya drink now.
Okay.
Soya drink.
Anyway, the point is that if you didn't have the mold, you couldn't produce the penicillin. But Fleming had sent some penicillium to Dr. H. Schmidt in Germany in the 1930s. Schmidt couldn't get it to grow, but even though the Germans didn't have a viable strain, other people in Europe did. So the Germans apparently did actually end up being able to get some penicillin, get their hands on some penicillin. So both sides kind of did end up having penicillin to use, supposedly. Now,
this is like Alan Turing being like, I sent computers out to everyone, and some of them were German.
This, bearing in mind, he discovered this before 1928, long before World War II.
I know. Yeah.
So in 1945, Fleming, Florey, and Chain, but not Heatley, were awarded the Nobel Prize in Psychology or Medicine. And in his acceptance speech, Fleming basically warned that the overuse of penicillin would lead to bacterial resistance. So even with his
Clever
early
man.
studies, he started studying bacterial resistance. He noticed that some bacteria were resistant to penicillin already when he'd started working on it. So in 1990, Oxford made up for the omission of Heatley from the Nobel Prize and awarded him the first honorary doctorate of medicine in its entire history. So bearing in mind, penicillin was worked on in Oxford University, so they gave an honorary doctorate to the man who had helped with it.
That's what you're going to
Nice.
get, Corey, if you keep up the SCI Guys.
That's what I'm hoping for.
They'll have to give you one.
Yeah.
They'll have to. They'll have
Do it
to.
without any of the work.
Yes, because none of this is work at all.
True.
If they give Luke a doctorate, that's
They'll give you
Oh,
a
man.
I'll give you
Hell
a
yeah.
knighthood
Race
for
to
putting
get a
up with me and
doctorate
Luke.
between me and Corey. Place your bets, guys.
So patents. You guys know what patents are?
Yeah.
Yeah, Luke, do you
We've
want
gone over them before.
Yeah.
Oh, that's loud.
Luke, do you want to briefly explain what a patent is?
It is a sort of like a license to be the exclusive person to use a certain technique for a set amount of time.
Yeah. So a technique or technology, you're the
you
only
discovered or someone you employ discovered.
Well, you don't even need to necessarily have discovered it, I don't think. You just need to be the first one to put the patent in.
But then they wouldn't give you the patent because it would be prior art or prior work.
Yeah. I mean, you could steal it, is my point.
Yes, and there have been some people who've tried to do that.
Yes. I suppose if you're going to be honest, yes, you need to have discovered it. So, yes, patents are basically a license to use a technology or technique. Now, the issue was that Chain thought that it was essential to get a patent, whereas Florey and some others thought it was not ethical to get a patent on something that was life-saving and revolutionary like this. And it ended up, basically, there was this whole issue of whether you can patent penicillin or not, because it was produced by a living organism. So generally, the view of Britain at the time was that you can patent a process, but not the chemical. So anyone can produce penicillin, but only people with the patent could produce penicillin under the specific
way of doing
it that they had created. So that's generally still lasts today. You can't really patent a biological chemical. You can't patent something that is produced naturally. You can only patent a way of producing it.
And that fact is the bane of every pharmaceutical company in the entire world. Because there's no point doing research into plants because you can't patent them.
Yep.
Very sad.
Genuinely, you can't patent.
Even though they're generally more helpful.
Well, you can patent a way of producing it, but you can't patent the plant itself. So we've discovered the
We've discovered penicillin right here, right now.
Oh my God, we should patent it.
So we've
Can't
covered
patent the chemical.
We've covered the discovery of penicillin, and in fact, like I said, penicillin is produced by the blue-green mold that grows on bread. So maybe we could try getting some of our own penicillin. I actually produced chemically penicillin for my final project in my last year of high school. Yeah, so I was making penicillin through a chemical process. Went terribly, didn't work, turned into black sludge most of the time. But we could potentially try and do some work with penicillin using bread mold. Why don't you stick around and maybe see that? It might come in
I think I've got some moldy bread around.
Yeah, it might come in. It might that might be coming in a few weeks. We don't know.
How about we infect James with something and then give him our homemade penicillin to see if it works?
I'm actually
You mean
open.
infect
I am
him?
open to that.
You mean infect James and then make him eat moldy bread? Sure.
Yes.
Okay.
Yes.
Fine.
Maybe
That
not
would make a
moldy
hell of a
bread,
video, wouldn't it?
We kill our friend.
Eating moldy bread to cure my illness.
And then spitting it out again.
Gone wrong.
No. Okay, so we've covered the discovery of penicillin. We've spoken about what happened after it was discovered in the kind of short term. But what is it what is the current state of penicillin? I've brought up briefly the topic of antibiotic resistance. So there are obviously some bacteria that are resistant to penicillin now because we just used it willy-nilly. I say willy-nilly, we use it for a lot of things. I mean, penicillin is kind of your standard broad spectrum antibiotic, and very few people are kind of allergic to it. So it's something that you can use on a lot of things. So how are bacteria becoming resistant to it? And then I'll get into a little bit of what bacterial resistance means for the world. So there's penicillinase, bacteria are sometimes able to produce penicillinase, which is basically just an enzyme that
Mayonnaise with penicillin in it.
no, you can get it at Nando's actually. So bacteria produce penicillinase, which is just an enzyme that can break down penicillins, and that can spread through different bacterial populations. So what bacteria can do, while they can only replicate through asexual reproduction, they split themselves into two. What bacteria can sometimes do is to share DNA between each other without reproducing. Yeah. So the only way that humans can really share DNA, the only way that mammals can really share DNA is by having sex and producing offspring. Whereas bacteria can kind of
Sharing
do it the
some
other way
DNA.
around, where they have sex with themselves to produce offspring, but they can share DNA so across a generation. So we can only share DNA
Wow.
down generations. Bacteria can do it across a generation. Which is
Fungus do that as well, I think.
Yeah,
Mushrooms do that,
yeah.
where they like one bit of the mushroom will because mushrooms can be gigantic. They can be like the mycelial mass underneath the ground can be gigantic, and then they send up mushrooms in different areas. And if a mushroom like encounters, for example, a type of thing that kills it, it develops a way of defeating that, and then it passes that information back through the mycelial mass.
So I think
Something along those lines.
a potential issue people might have with that is that in those kind of mycelial structures, that you could kind of say that's one organism rather than different
yes.
members of the same species.
Of
Whereas
course.
what bacteria can do is actually do it
Tell each other.
completely different members. So that's the equivalent of me giving James a gene for poor eyesight, perhaps.
I've got enough, thank you.
Okay, that's fine.
Wow. So it's like our internet. It's like where we can share ideas with each other.
Yeah, it's the bacterial internet.
Hey,
bacterial
this-
internet.
This idea I came up with is freaking sick.
So
You
this
try it out.
process is called conjugation, and it's effectively the bacterial equivalent of sexual reproduction. But it doesn't involve actually producing a new organism. It's just sharing information. So another way that they could do it is through altered bacterial structure. So as I said, penicillins attack the cell walls of bacteria, but what they can do is just slightly change the structure of those cell walls so that the penicillins can't bind to it, which obviously incapacitates penicillin in its attempts to destroy the bacteria. And another system that they've got is penicillin removal. So what they can do is just develop a system to get rid of penicillin. You put the penicillin in the bacteria and it says, I'm not having that, ships it right out. So those are just 3 ways that bacteria can have a resistance to penicillin. And actually, bear in mind that a lot of these kind of, if it's based on just a couple genes, they can spread it amongst the population. So it does. If one bacteria comes up with a way to get rid of penicillin, it can spread it amongst the entire culture.
Yeah.
Potentially. And the reason that antibiotic resistance is such a big issue is because we're using antibiotics for a lot of things. What we do is we end up generating superbugs. So it's effectively, let's say you use penicillin until that stops working, and then you use a different antibiotic until that stops working. At some point, you're going to run out of antibiotics. And the issue right now is finding new antibiotics that can kill.
Yeah, we've not found one for
bacteria.
like 20 years or something, have we?
Yeah, I mean, I don't know that off the top of my head, but I mean, the fact is that finding new, go ahead and have a look. But the fact is that finding new antibiotics that can tackle the super bugs that we created is an issue. And if we're not careful, we're going to get to a point where antibiotics are basically useless against the biggest diseases. Because
So this is called the discovery void,
Mhm.
and it refers to the period from 1987 until today when the last antibiotic was
ongoing.
discovered, which means that it's been 33 years since we discovered an antibiotic.
Whoa.
that's insane.
That seems like it's too long.
Running
It's
out
kind
of
of
time.
it's kind of like a fossil fuel situation where we
have run out, it seems.
Well, yeah, I mean, what I mean is with fossil fuels, we use them kind of willy-nilly, and then we realize, oh no, this problem is happening, and we're in it right now, and we need to start slowing down to make
Where
sure
do
that it
most
doesn't
of the
get worse.
antibiotics go, Corey?
To animal farms.
To animal
80% of
agriculture.
antibiotics.
I
Not
didn't
to
know where you were
us.
going with that. Yeah, so that's actually something they use in sort of factory farms. Effectively, what happens is because animals are in such close quarters, they're obviously spreading diseases amongst each other. The easiest way to stop an outbreak, just willy-nilly
Pump them full of antibiotics.
pump them full of antibiotics.
Yeah.
If you basically administer antibiotics to your entire herd of cattle, or all of your chickens that are in their battery cages, then
Which I frequently do.
yes, well, Luke does that every weekend. That's why he's so
Every
busy. But if you do that, then you don't need to worry about infections breaking out because you've basically just killed all the bacteria anyway.
Yeah.
The issue is that you basically just create superbugs that are resistant to all of these antibiotics. But that is the end of the show now. We've spoken about antibiotic resistance and the discovery of penicillin. But now it's time for my favorite segment, and that segment is the Quickfire Quiz round.
Dun, 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 dun.
Jamp, what is your buzzer?
Quack.
Luke, what is your buzzer?
Oh, I
Okay.
like that one.
Fantastic. In what year was Alexander Fleming born?
Quack.
In BL.
Quack.
Okay, Jamp, I'm going to go with you.
Oh, shit. 133 BL? No, it's...
I'm sorry, Jamp. You said it. That is not correct. Luke, you can take the steal.
113 PL.
Ding, ding, ding. That is correct. It is 113 BL. Now,
I second guess myself.
question number 2. Guys, get ready. There's still everything to play for. Okay. Which chemical does penicillin target that makes up the bacterial cell wall?
Can't remember.
Luke, you tried
Forgot
really hard
that
to
one.
remember this one.
Oh, did I?
Oh.
Oh, dear.
Oh, for God's sake.
Oh, dear. Oh, come on. Oh.
Eosinophilate.
I'll take a guess at this point.
Mitochondria.
Was it what James said?
No.
Cellulose.
I'm afraid it's peptidoglycan.
That's the one I was
Of
thinking of, but I
course,
couldn't remember how to pronounce it
peptidoglycan.
at all.
Classic peptidoglycan.
That's what I meant by phyllosellophyllate.
Okay.
Yeah.
Yeah.
Last question.
Okay.
Where would you be able to find the enzyme lysozyme?
Oh,
I
quack.
don't know.
Jamp?
In your nose and in your tears.
Okay, that's right. In your tears.
In
Ding,
your
ding,
snot.
ding.
Yeah.
You can find it in your tears.
Ah.
Yes. Okay, that means we're at 1-all. So this is the tiebreaker. In what year was penicillin first discovered? Luke?
1928.
Ding, 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 ding. That is correct.
I
It
truly
appears
hate you.
that Luke is the winner of this week's Quickfire Quiz round. I sure hope someone is keeping score because we are not. Let us
If I
know.
was better with dates, I'd be winning more of these.
But that is this week's episode. Congratulations, Luke. I am sorry, Jamp. Maybe you have better luck next time. Thank you for watching. You can find the full references for this episode in the description. Subscribe for new episodes every Sunday. And why not leave us a nice wee comment? You can support the pod at patreon.com/SCI Guys, or you can find and contact us at SCI Guys Pod on Twitter, Facebook, Instagram, and YouTube. Or send us an email at sciguyspod@gmail.com. I am not Corey everywhere.
I'm Jim Everywhere.
I'm Luke Cupboard everywhere.
Goodbye.
Goodbye.
Bye.